My name is Gernot Walko. I'm from Austria and I'm currently working as a senior postdoc in the lab of Professor Fiona Watt here at the Center for Stem Cells and Regenerative Medicine. I obtained both my undergraduate and postgraduate training from the University of Vienna, where I also did my PhD in the lab of Professor Gerhard Wiche. During my PhD thesis, I was investigating the molecular mechanisms underlying rare forms of a severe skin blistering disease called epidermolysis bullosa. And it was during that time that I developed a strong interest in skin biology, especially in understanding how the epidermis, which is the outermost protective layer of our skin, is maintained throughout adult life. So following an appointment as university assistant at the University of Vienna, which allowed me to combine my passion for research with my other great passion for teaching, I eventually moved here to the United Kingdom to join one of the world-leading laboratories in the field of skin biology. My research here at CRCRM focuses on the interactions between human epidermal stem cells and the local microenvironment, which we like to call the niche. So I'm interested in understanding how these stem cells integrate the numerous signals they receive from the local microenvironment with their own cell intrinsic gene regulatory networks in order to make the decision of whether to remain a stem cell or to commit to terminal differentiation. And I hope that by understanding how this stem cell state is maintained, we will be able to provide improved protocols for rapid expansion and long-term culture of human epidermal stem cells for regenerative medicine purposes.
Hello, my name is Ruta and I'm working with HIPSI phenotyping group in Fiona Watt lab. I came from Lithuania, where I've done my undergrad and postgraduate studies in molecular biology and genetics. After my studies, I joined the lab in Wellcome Trust Sanger Institute in Cambridge, where I worked with the Knockout Mouse project, where I was involved in generation of knockout vectors and quality control in transfected mouse embryonic stem cells. Over there, I, for the first time, tried to maintain human iPS cells, and that's how I fell in love with them. And that's how I joined Fiona Watt lab for the open position to be a research technician in HIPSI project. In HIPSI project, we characterizing hundreds of induced pluripotent stem cell lines derived from healthy donors and patients with selected disease. We developed 96-well endpoint assay to, based on the different concentrations of fibronectin to see the cell lines and their morphology, proliferation, and clumping. We believe that this assay helps to select good donors from panel of iPS cells, and it can be used for disease modeling and drug discovery. For the future, we are planning to modify this assay for feeder-free cell lines, and also we're thinking to introduce in the assays chemical stimuli and microenvironments.
Hi, my name is Arsham Ghahramani and I'm a joint PhD student based here and at the Francis Crick Institute. I joined the Watt lab earlier this year, and my research mainly focuses on the epidermis, which is the uppermost layer of the skin. I'm originally an engineer by training, so in the past I've been involved in a variety of engineering research projects, one example being that I helped develop a medical injection device for pharmaceutical industry. Since joining the Watt lab, I've really benefited from the expertise that we have here in cell biology and the skin, and a major theme of my PhD is taking that expertise and combining it with the computational expertise that I've gained in the past. So my research is interested in understanding how cells within the skin communicate with each other to maintain a population of stem cells which aids the regeneration and healing in the skin. I'm achieving this through the use of a recently developed technique called single-cell RNA-seq. This is a technique that allows us to measure gene expression at the single-cell level. This technique requires a lot of computational analysis and mathematical modelling, which is where my previous expertise comes in. But the advantage of using this technique is that we can look at gene expression at single cell resolution, which hasn't been done in the skin before. I hope to take the knowledge from my research forward in the future to use the new knowledge that we've developed about how cells communicate in the skin to then help improve the regeneration of the skin and our understanding of healing.
Hi, my name is Ashley and I'm a PhD student here in the Centre of Stem Cells and Regenerative Medicine. Before I joined Professor Watt's lab, I completed my undergraduate degree in cell biology at Durham University. During this time, I spent a year developing a flow cytometry-based assay for the rapid detection of dendritic cell and monocyte disorders using patient blood samples. Following this, I came down to London to complete my Masters of Research in Biomedical and Translational Sciences. The programme allowed me to rotate between 3 different labs, and I decided to take this opportunity to expand my skill set and explore a wide range of fields, including spinal cord injury, acute liver failure, and iPS cells. This brought me to do a PhD here in cancer research. The aim of my PhD is to correlate genetic changes with changes in cell behaviour in squamous cell carcinomas of the head and neck, with a particular interest in inactivating Notch1 mutations. I'm currently working on inserting the active NICD portion of the Notch1 protein into patient-derived stem cells. I'm utilising an inducible lentiviral system and high-content screening in order to identify dose-dependent changes in cell characteristics which are associated with Notch signalling. Outside of the lab, I also believe in the importance of public engagement in science. As such, I'm a STEM ambassador and part of the Voice of Young Science venture. I spend a lot of time visiting schools and youth centres in order to increase enthusiasm and widen participation in science. Following my PhD, which I currently have 2 years remaining, I'm confident that I wish to continue a research-driven career path. In particular, after experience in a number of different fields, I wish to remain within cancer research. Of particular interest, I enjoy translational projects, and if there was a way to incorporate my enjoyment of public engagement, perhaps even through lecturing or direct patient contact, that would be a huge bonus.
Hello, my name is Ayelen. I come from Spain. I have a Bachelor in Chemical Engineering and a Master in Material Science and Technology. I have been developing my career in biomaterials for regenerative medicine for the last 4 years in different universities of Europe, in Santiago de Compostela, in Ireland, in Denmark, and currently I'm living in London. I'm working at the Centre for Stem Cells and Regenerative Medicine as research assistant under the supervision of Professor Fiona Watt. My project consists in developing an in vitro dynamic model to understand the influence of epidermal-dermal junction topography in stem cells behavior. In the future, I would like to continue my career in materials for regenerative medicine.